E agora eu vou convidar o Dr. Gabriel Lamothe. O Dr. Gabriel Lamothe, ele é um mestrando do laboratório do Jacques Tremblay, e ele é o mestrando que obteve a maior bolsa de pesquisa de todo o Canadá. Dr. Gabriel Lamothe, it's your turn now. Welcome.
Thank you for having me. Let's see if we can get it started. Hi, my name is Gabriel Lamothe, and today I'll be discussing the detection of SARS-CoV-2.
Yeah, that's much better.
Is the sound better now? Using CRISPR technology. Oh, perfect. Namely, the test that we've been developing in Dr. Tremblay's laboratory. Studies have confirmed that many individuals infected with the SARS-CoV-2 virus are asymptomatic carriers of the virus. This is problematic in that the pandemic might be growing without adequate supervision. Simply focusing the testing on individuals with symptoms as opposed to entire populations is not enough. To better contain the virus until a large-scale distribution of an effective vaccine is underway, large-scale and continuous testing is mandatory. The current diagnostic pipeline for detecting SARS-CoV-2 is a three-step process. The first step is to obtain nasopharyngeal swabs from patients. The next step is to isolate the total RNA from said samples and provide the RNA required for this third step, which is to use real-time quantitative PCR machines. The RNA is first reverse transcribed into DNA and then amplified. A dye is added to the amplification reaction to signal the presence of DNA strands. After a certain number of amplification cycles, a positive result will give a strong enough signal that the machine flags it as containing the original virus. Now, this process is problematic for a few reasons. The first is that real-time PCR machines are extremely expensive. Most can range from $15,000 to well over $90,000 US. Given that it's a fairly specialized piece of equipment and that not all molecular biology laboratories need one, some get along just fine with a regular PCR machine, which costs a much more reasonable $5,000 US. As such, widespread testing for the coronavirus using these machines isn't ideal. Acquiring the large number of machines required for widespread testing is a feat in and of itself, and is likely one of the contributing factors for the sometimes long wait times for results. Many reports have stated that it can take 3 to 24 hours or more to obtain results in a clinical diagnostic laboratory. Over here in Canada, Ontario reports wait times of up to 4 days to get results. High-density metropolitan areas like New York are also subject to long lines for testing, and then a time to results of about 7 days for some clinics in that city. The creation of new types of detection tests is therefore an important step in curbing the spread of this virus. An ideal test needs to follow the World Health Organization's criteria for assured. That is, the test needs to be affordable for as many people at risk of infection as possible. Tests that require extremely expensive reagents or machinery are not ideal. The test must also be specific to avoid false negatives. That way, anyone who could potentially spread the virus can be identified and put into quarantine. The test must be sensitive so that few people who don't have the virus are mislabeled as having it and treated improperly. The test must also be user-friendly. That is, it must be simple to perform. And it must be rapid and robust. That is, it must give a time to results that is quick enough to make the most use from the results. Situations where it can take up to 7 days to obtain results are clearly far from ideal, as by the time the patient receives the information, they might have infected many others. The test must be equipment-free as much as possible. Tests that require too many rare and expensive machines aren't ideal for many countries. Finally, the test must be deliverable to those who need it. The discovery of Cas13, or C2C2, was revolutionary in the field of molecular detection. This class II, type 6 CRISPR effector is similar to the protein Cas9, off of which base editing and prime editing were designed. This protein is also a member of the CRISPR family. Unlike Cas9, however, Cas13 can be programmed to target RNAs as opposed to DNAs. By binding to its CRISPR RNA, Cas13 is primed to target extremely specific RNA sequences. After locating a target sequence, the Cas13 protein is activated. This causes the protein to begin to cleave everything in its surroundings in an event called collateral cleavage. It cleaves its original target and then moves on to start cleaving all surrounding RNAs. Researchers realized that this collateral cleavage could be used to turn the protein into a switch for a detection test. Once the target molecule is present, the switch is flipped, the Cas13 activated, and all surrounding RNAs are degraded. By tagging certain RNAs in the solution with a fluorescent molecule and a quencher, it would therefore become possible to resolve a signal. Researchers therefore started to develop a detection test based on this protein for viruses, bacteria, and human diseases such as cancer. However, they soon noted that there were certain limitations to the protein and that its limit of detection was far from sensitive enough for their needs. Recently, certain tests have been designed using only Cas13. However, these tests are less sensitive and tend to use a different Cas13 protein. Researchers at the Broad Institute decided to supplement the protein with a few others that would amplify the target RNA. To this end, Dr. Zhang and his team created a new test called Specific High Sensitivity Enzymatic Reporter Unlocking, or SHERLOC for short. This test is based on the reverse transcription of target RNA into DNA. Afterwards, the DNA is amplified at a constant temperature by using a process called recombinase polymerase amplification. This technique amplifies a small amount of DNA at a constant temperature, unlike the more commonly used PCR. The incorporation of this step also means that the ensuing detection test can target DNA. Since the RPA reaction simply amplifies a determined section of DNA using two primers, the initial genetic sequence can either be RNA or DNA, so long as reverse transcriptase enzymes are present for RNA. The amplified sequences are then transcribed using a T7 RNA polymerase to create vast quantities of the target RNA. This then allows the Cas13 to have a far larger pool of target molecules, which can activate the enzyme. Once the enzyme is active, it does what we discussed before and starts to cleave all surrounding RNAs, including reporter RNAs, which give off the easily detectable signal, which makes the test work. In their initial version of the SHERLOC test, two different reporter molecules were used. The first used a fluorescent molecule as well as a quencher, and it was able to produce a lot of fluorescence after cleavage of the RNA. The other was designed to interact with a few antibodies, and when it was cleaved, it appeared as a distinct second band on a strip that was quite similar to a pregnancy test. While this test was good in many ways, it had certain drawbacks. At the beginning of the pandemic, we attempted to replicate it and immediately had a few issues. The biggest issue by far was that some of the materials required for the test to work were only available in extremely limited supply. The kits required to perform RPA reactions, for example, took approximately 2 months to arrive, and even then, we only received enough materials to perform 200 tests. This clearly wasn't going to be enough to produce sufficient tests in the future. Additionally, the reporter that used a pregnancy-style strip was remarkable and seemed to be very useful. However, since it was too difficult to acquire in sufficient quantities, we decided not to use it and to instead focus our efforts on using the fluorescent-style reporter RNA. Since the original version of the test could not be used by us in the way that it was intended, we decided to redesign it. In redesigning the test, we had several options and routes that we could take. We knew that we wanted to keep on using Cas13 because we had a large supply of this enzyme due to an ongoing collaboration with Dr. Alain Garnier's lab. We also knew that we wanted at all costs to maintain the isothermal nature of this test. One of the biggest advantages to SHERLOC was that it didn't require large amounts of expensive equipment. Simply by using a heating block or something of the sort, it was possible to get a visual readout. We therefore decided to employ loop-mediated isothermal amplification, or LAMP for short. This amplification strategy was fairly well documented and easy enough to perform. Based on the use of 6 primers, this technique could rapidly amplify large amounts of target DNA. Unlike both RPA and PCR, however, LAMP doesn't create identical amplicons of a discrete size. Instead, the 6 primers can interact to create far larger molecules of a repeating sequence. Initially, it was a little difficult to incorporate this isothermal amplification strategy into our test. As you might remember, the step following RPA in the original SHERLOC test was one that produced lots and lots of target RNA. This production of target RNA was only possible because the RPA step added a tag that let the T7 RNA polymerase bind to the DNA and begin transcribing it. However, no one had seemingly ever done that with LAMP, and since this new technique used 6 primers, 2 of which looped back on themselves to create weird dumbbell-like structures, it was difficult to figure out exactly where to incorporate this tag. After a while, we decided to include the tag right in the middle of one of our looping primers. This has consistently given us very strong results. We were therefore able to retrofit the original Sherlock test with a new amplification strategy, all the while still benefiting from Cas13's specificity and signaling. The new test is therefore using RT-LAMP as opposed to RT-RPA. Of course, should we become interested in targeting DNA in the future for either certain types of viruses or certain bacteria, this test would be readily applicable to that as well. We also decided to use a fluorescent reporter RNA instead of the pregnancy-style one that we spoke about previously, because it seemed to be the most readily available material. Since it doesn't take any particularly advanced machinery to make it work, we were happy to choose this one. Now that you know how the test works from a theoretical standpoint, let's discuss it from a more practical point of view. For now, the test requires that we start with RNA that was extracted from a virus using a commercially available kit. 1 microliter of this solution is transferred over to the tube containing the RT-LAMP reaction. Ideally, in the future, we would like to modify the test so that it becomes possible to directly add a patient's saliva to it and continue on from there. That would noticeably cut down on the labor required to obtain each reading, cut down on the overall costs, and make it much faster for the results to come in. After transferring 1 microliter of the RNA extract to the first tube, the tube must then be incubated at 65 degrees Celsius for 30 minutes. After that, 1 microliter is transferred from the first tube into the second, which contains the T7 and Cas13 solution. After tapping the tube a few times to mix it and microcentrifuge it down for a few seconds, all you need to do is incubate that tube at 37 degrees Celsius for 30 minutes. At that point, all of the reactions are done and the results are ready to be read. By exposing the tubes to a wavelength of approximately 490 nanometers, it's possible to easily detect which samples contain SARS-CoV-2. Those ones will fluoresce a bright green. Those that didn't contain any of the viral specimens of interest will be completely colorless and indistinguishable from a tube filled with a little water. I'll show you that later. Now, it's worth noting that the DNA and RNA sequences that we have used in this first version of our test were all previously published by different groups. As such, we do not believe that they will result in a positive signal when in the presence of a relevant RNA. This is particularly important because your saliva is full of RNA. The extraction process for the virus does indeed result in viral RNAs being captured, but there tends to be a whole lot more human and bacterial RNAs too. In addition, other coronaviruses that are much less dangerous are fairly common in the human population. If our test gave positive signals every time someone came in with a common cold, then it wouldn't be very effective. As such, we have based ourselves on previously described templates to try and avoid this as much as possible. In designing this test, we tried to make sure that it wasn't overly reliant on complicated machines. We wanted the entire test to be easy to perform using only a few common laboratory apparatuses. As a result, with this current iteration of the test, all we need is 1 machine that can heat PCR-style tubes to 65 degrees Celsius, and one that can heat the PCR tubes to 37 degrees Celsius. We also need 2 either P2 or P10 pipettes for reasons we'll get into later, along with their associated pipette tips. A small microcentrifuge capable of taking PCR tubes is also required just to make sure that the reaction mixtures don't touch the top of the tube when you're opening them and potentially contaminating all of the surrounding tubes. Finally, a machine capable of giving off ultraviolet wavelengths is necessary. In this picture, you'll see the machine I've been using. It's most commonly used to visualize gels in a molecular biology lab, but it does a really good job of visualizing our positive samples in our test. For the 2 incubation periods, several different options are available. I've been using an old thermocycler to date, as I find it does a really good job of evenly heating my tubes. It's worth noting here that this means that even old and run-down thermocyclers that no longer do a very good job of cycling between temperatures can be readily used in this test. Unlike the current RT-qPCR gold standard that requires specialized real-time thermocyclers, here any old machine can be used. It's also possible to use other common laboratory apparatuses, such as block heaters, that have an adapter for PCR tubes. Please note that in this picture, the block heater has an adapter for 1.5 milliliter microcentrifuge tubes, so the true adapter will look a little different. I've also used incubators to great success. Even bacterial incubators that operate at a steady 37 degrees Celsius can perfectly suit the needs of this test. So long as the tubes are readily exposed to the ambient temperature, then positive signals should have no difficulty in resolving. Please note, however, that when using incubators, it's better to have the tubes exposed to the air. That way, they don't waste time as their rack slowly begins to heat up. Water baths are one final option that you can choose. Personally, I tend to avoid this option because I find the risk for cross-contamination between samples to be much greater and therefore unideal. However, I have tried to perform RT lamp reactions with this system, and so long as you're careful, it's very possible to do it. Once the reactions have been completed, it's possible to use various different machines to visualize the tubes depending on the materials your laboratory has. Personally, my machine of choice is a gel UV emitter. These machines, which are more commonly used to visualize agarose gels, do a very good job of safely showing which tubes are fluorescent and which aren't. Black lights can also be used for this test, however, I have found them to be slightly less desirable. Since the reporter RNA best absorbs wavelengths of 490 nanometers, I have found that black lights which produce wavelengths of 390 nanometers to be less favorable for this test. For them to work, it's better to visualize the tubes in a dark room. In both cases, the machines should really be placed in a darker room away from excessive light. A little background light seems to have less of an effect in the case of the gel UV light, however. This picture, seen on the left, was taken using a generic iPhone when the tubes were placed on the gel UV light. The lights in the room were only partially turned off, resulting in lots of ambient lighting, even if there were no lights directly pointed at the machine. Given the strong and even radiation that the tubes received on this machine, it's easy to determine which samples were positive and which were negative. In the case of the black light on the right, it's clear which samples are positive and which are negative, of course. However, the fluorescence is not even, and it's a little bit harder to detect by the naked eye. In this case, the camera on the iPhone took a very good picture and made it easier to visualize the tubes overall. Now, there are several important considerations to be had when using this test. For it to work properly and not give either false positives or false negatives, it's important to first designate proper working stations for each section of the test. It's also necessary to follow the predetermined reaction times without varying, otherwise the results might start to differ slightly. Finally, those handling the test must be careful not to contaminate any of the tubes with RNases. The first consideration to separate working areas is extremely important because lamb is excessively sensitive. This technique is known to produce lots and lots of concentrated target DNA. We, as well as others, have noted that when we open tubes that completed the lamp amplification in the same area that we originally used to set up the reaction, it's possible to contaminate the working area with aerosols containing the product. If you continue to set up the lamp reactions in those areas afterwards, then you run the risk of contaminating your new tubes with amplicons from the previous reactions. By touching your desk or various other contaminated surfaces with your gloves, you run the risk of artificially seeding your reaction mixtures that contain patient RNA with no SARS-CoV-2, and therefore producing positive results when there really shouldn't have been any. By separating the areas in which you set up the lamp reactions and those in which you use the amplified sequences, you run less of a risk for this to happen. It's also necessary to have designated lab coats, pipettes, and other such materials for each working station. If those in workstation 2 become contaminated, you don't want that coming back into workstation 1. Please note that this is much less stringent when bringing materials from Workstation 1 to Workstation 2. Since it's the RT lamp reaction that runs the greatest risk of contamination, bringing material, for example, gloves, from that station to the next should not be a problem, provided that the gloves are still relatively clean. Please note that this situation of contamination via aerosol is of much less importance during the second stage of the test. As we discussed previously, the Cas13 protein we are using requires a relatively large amount of RNA to be functional. At the present time, we are not concerned in the least that an aerosol contamination from LAMP might produce false positives of our Cas13 reactions, as we have yet to see a single situation where this has happened. The second consideration of note is that the reaction time must be followed. A small discrepancy can be tolerated, that is, 1 minute more or less doesn't really seem to have a large impact on the test. However, more than that can start to become problematic. In the case of the RT lamp reaction, external teams, as well as us, have seen that by doubling the reaction time for the lamp, false positives can arise. As you can see here in both pictures, a successful lamp reaction can also be visualized on an agarose gel. The successful reactions are characterized by the presence of several large bands of varying sizes. However, when the lamp reactions go on for too long, it's possible to begin amplifying samples with nothing in them. For example, on the left, you'll see a gel that was taken from an article in which at 30 minutes, the wells containing reactions with 10 copies of the target viral RNA and no copies of the target viral RNA had no bands. In the case of the gel on the right, a gel that I made, 3 of the wells contained reactions that had no target RNA to speak of. Only the N fragment well contained target RNA. At 30 minutes, these negative controls did not demonstrate any amplification, as we expected. However, at 60 minutes, both the published gel and mine started to give amplified products. In their case, the 10 and 0 copies both started to be amplified, and in my case, one of my negative samples started to get amplified. I've performed several experiments so far with 30-minute incubation times, and so far have never gotten false positives. That's not to say that it's entirely impossible, simply that it's very, very unlikely. As a side note, this is a good time to mention that changing gloves frequently when performing the first reaction is a good way to avoid these kind of unlikely false positives. After prolonged use, gloves might become contaminated with small amounts of viral RNA, and that could potentially lead to false positives, even if we're only incubating at 30 minutes. The final consideration to look out for is the presence of RNases. For those of you who might not be familiar, RNases are enzymes that cleave all RNAs they come across. Both steps for this test are sensitive to RNases. The initial RT lamp begins its amplification off of RNA, so the presence of RNases could be very detrimental and therefore lead to false negatives. The second step of the test, the T7 and Cas13 step, uses an RNA reporter to signal positive tubes. If, while you're using the test, you start to introduce RNases, then you run the risk of getting false positives. RNases are extremely difficult to remove once they're introduced into a system, so the best means of protecting your test is by using prevention. Since your hands produce lots of RNases, it's mandatory to use gloves at all times when working with the test. In doing so, you will prevent your body's natural RNases from degrading the initial SARS-CoV-2 RNA and protect the fluorescent reporter at the end. Gloves must also be changed whenever they come into contact with skin, hair, or other frequently manipulated objects, such as doorknobs and personal devices. The use of RNase and DNase-free filtered pipettes is also important for this test. By using these kinds of specialized tips, you can make sure that any possible contaminations you might have in your pipettes doesn't have a chance to contaminate your test. In areas where the risk for RNase contamination is fairly high, it might be worthwhile to treat all surfaces or tools with an RNase removal solution. These kinds of solutions can degrade RNases that might otherwise stick to your gloves and contaminate your samples. Please note, I haven't had to use this solution very often. So long as you maintain a clean working schedule, you should be fine. The second component for the test, the one containing T7 and Cas13, is also designed to be able to tolerate a certain level of contaminating RNases. There is an RNase inhibitor added to the reaction mixture to prevent the degradation of the reporter in case the original SARS-CoV-2 RNA wasn't completely clean, or in case contaminations occurred while you were preparing the test. That said, the presence of excessive RNases can still fully activate the test. Seen here on the right, 1 microliter of very concentrated RNase A is capable of causing the test tube to become fluorescent. It's therefore important to take the required
Gabriel, on t'entend peu?
test that can be widely distributed while also meeting this limit of detection is fully functional for curbing the spread of this pandemic. While RT-qPCR is much more sensitive than this, its improved limit of detection might not necessarily be that beneficial. Since this technique can pick up RNAs freed by infected and dead cells after the infection has run its course, this form of screening can cause individuals who are nominally SARS-CoV-2 RNA positive, but aren't actually infectious, to be isolated unnecessarily. As you can see here on our left, our detection test is able to detect samples that have 100 copies per microliter, as was stipulated by the model. A more recent comparison between the different primers that we've been using for LAMP has demonstrated that one version of our test can detect samples containing only 80 copies per microliter, while the other can only detect samples containing 1,000. Moving forward, we're likely to focus our efforts on the first primer set to ensure that we meet the limit of detection that was stipulated by the model. As a final note, we're happy to report that our test works not only on fragments of the SARS-CoV-2 genome, it also works when exposed to the virus's full RNA profile. Initially, we began the development of our test by using fragments of the virus's genes that we had transcribed ourselves in the lab. Recently, we've used the test on actual SARS-CoV-2 viruses and have obtained the expected results. As you can see on the left, the first section of the reaction, the RT-LAMP reaction, works just as expected on the viral RNA. The top left wells that were run on the agarose gel clearly demonstrate a strong amplification. Below them, 3 reactions were performed with cells that had not been cultured with the virus. The viral RNAs were created by first isolating the viruses from a patient. These viruses were then cultured in Vero E6 cells, and after a few days, the culture medium was treated to extract any and all RNAs that were present. The viruses were therefore destroyed and their RNA was obtained. When the LAMP amplicons that had amplified the real virus were transferred over to the second stage of the test, they gave the expected strong fluorescent signals. At the moment, what remains to be done is to use our test on samples that have been confirmed positive or negative by RT-qPCR, the current gold standard in SARS-CoV-2 detection, and see how our test compares. This will give us a better indication of what our test's sensitivity and specificity are. Just to wrap up, I would like to thank Dr. Gary Kobinger, Dr. Alain Garnier, and Dr. Guy Boivin for having helped to provide finances or materials for this project. I'd also like to thank Dr. Tremblay and everyone in his team for having helped to provide certain insights as I was redesigning this test. Finally, I'd like to thank Dr. Marcello for having helped to organize this entire meeting, and the Canadian Institute for Health Research and the FRQS for having helped finance me as I pursued this project.
Okay, so that was my presentation. Yeah.
Thank you, Gabriel. I'll take the time just to make to summarize it the best I can here for the non-English speakers. Thank you very much for your presentation.
Thank you.
Okay, then Vou fazer um resumo, assim como eu tentei fazer com a apresentação do Dr. Jacques. Deixo bem claro que é um resumo de uma apresentação que carrega tanto conteúdo e tanta experiência de trabalho, não pode ser considerado assim uma experiência, uma como assistir a apresentação. Então, se vocês tiverem a oportunidade de voltar ao YouTube, pesquisar melhor os temas, de repente assistirem uma velocidade mais lenta, talvez possa ser bom para todos vocês terem uma experiência melhor com isso. De toda forma, eu vou tentar fazer um resumo do que o Gabriel falou. Ele mostrou o desenvolvimento da Covid, que todos nós estamos acompanhando. De uma forma geral, o teste atual se faz através de uma máquina de PCR em tempo real, e esse é um processo muito caro que demanda o processo em si não é caro, mas a obtenção das máquinas é muito caro e elas não são bem distribuídas no globo, ou seja, não são bem distribuídas em todos os lugares que necessitariam fazer esse teste. Assim, surge a necessidade de se criar um novo estilo de teste para que possa ser, de uma forma mais geral, aplicado em todo o globo e acessível a todas as pessoas, como recomendado pela Organização Mundial da Saúde. A descoberta da Cas13, ou C2C2, que é o que ele ressaltou, torna importante porque ela faz mais, torna esse processo mais fácil e mais importante, uma vez que ela faz o mesmo processo que a Cas9 consegue fazer com DNA, só que ela faz com RNA. E dado essa característica de fazer com RNA, é possível isolar o RNA dos vírus para desenvolver um tipo de teste. E foi isso que eles fizeram. Alguns testes usando a Cas13 foram desenvolvidos, mas não foram muito seletivos, ou seja, não responderam muito bem a apenas aquele tipo de vírus, ou geraram algum tipo de falso positivo. Eles tentaram, então, desenvolver o estilo de teste que eles chamaram de Sherlock, que é um teste específico de alta sensibilidade enzimática, e esse tipo foi desenvolvido com 2 tipos de moléculas usadas durante esse processo. Tentaram manter esse design do Sherlock, atualizando ele com uma amplificação isotérmica, que é o que ele chamou de LAMP, e assim fizeram o teste para a COVID usando o Cas13A e esse Sherlock LAMP, essas tecnologias. Basicamente, o teste consiste em tirar algo que teve contato com o vírus, alguma amostra que teve contato com o vírus, passar pelo processo de amplificação isotérmica e incubar ela por 30 minutos a 65 graus Celsius. Daí tira-se 1 microlitro dessa amostra e coloca esse microlitro em uma solução que contém a Cas13 e mantém a solução por 30 minutos em 37 graus Celsius. O resultado será mostrado através do uso da luz fluorescente, que vai mostrar a cor verde em caso de positivo. Tivemos alguns desenhos dos slides dele, que onde é possível ver claramente esse procedimento do teste. É importante analisar o final do teste em um lugar onde não haja interferência de outros tipos de outras fontes de luz, porque poderia causar algum tipo de interferência na leitura do resultado final. De modo geral, o objetivo do teste é evitar que ele responda a outros tipos de coronavírus, já que existem outros tipos de coronavírus que não causam nenhum tipo de infecção tão letal ou tão ruim quanto o atual tipo de coronavírus que nós estamos enfrentando essa pandemia. O teste deve ser mantido o mais simples possível e, por isso, eles tentaram minimizar o uso de aparelhos. Então, ele mostrou na sua apresentação quão simples, relacionado ao estilo de teste atual, o teste que eles propõem pode ser. Então, a grande maioria dos aparelhos que ele mostrou são aparelhos comuns de laboratórios e não seria necessário uma máquina de PCR como é o estilo atual, dado que essa máquina é cara e tem pouca distribuição. Então, os aparelhos usados poderiam ser incubadores, aquecedores de blocos. Outras sugestões que ele faz para o uso desse teste é separar em diferentes áreas de trabalho cada teste ou cada conjunto de teste. Seria importante também seguir de forma estrita o tempo das reações, pois erros maiores que 1 minuto podem levar a falsos positivos, como eles observaram. E, finalmente, evitar contaminações de outros RNA vindos de fontes externas durante o processo de testagem. De uma forma geral, o teste pode ser usado com exemplos de RNA de vírus, numa forma geral, não precisa ser só para Covid-19, como é o caso da situação atual. Eu acho que eu fiz um apanhado bem geral dos dois e passo a palavra agora para o professor Jacques.